Ebola virus, Zika virus, West Nile, dengue. These are just a small number of infectious diseases that have reemerged into the public health sphere in recent years. Because of a number of factors, climate change, increased urbanization, or simply evolution, the need for new and innovative detection techniques is never likely to go away, as new pathogens emerge or old pathogens change to confound our current detection systems. But how can researchers work on building these detection systems without putting themselves and those around them at risk of infection? More to the point, how can most researchers even handle these pathogens when only a handful of labs in the world are capable of housing them safely? My name is Douglas Holmes, and my lab is working on a solution. Using synthetic biology, we can create DNA sequences that are the same sequences as those found in infectious diseases, but modified so that they can't produce protein, rendering them essentially harmless. We can then take these sequences and insert them into less pathogenic organisms and use them as a model surrogate. My lab has already used this technique on Ebola genes inserted into E. coli, which is a common laboratory bacteria. We were then able to detect these sequences using DNA amplification and are currently developing a DNA probe that will bind to these sequences inside the E. coli cells and glow, or fluoresce. This process is called fluorescent in situ hybridization, or FISH for short. Now, what does all this have to do with detection techniques? Using a specialized FISH probe, we can create a platform that can detect multiple diseases simultaneously in under an hour using no specialized training whatsoever. All that's required is a drop of blood or a cheek swab, something easily collected at the point of care, that's then placed on a test strip. My lab has already used the method behind this platform in our lab, but until now we've been unable to use it on pathogens because of the danger they pose. Until now. Using model surrogates like the one that we have designed and are currently designing more of, we can test these probes and build this platform without posing a risk either to myself or to public health at large. Thank you for your time, and I hope you find this research as interesting as I do.